Hi everyone, welcome back to another video. So today we're just going to do a short and sweet practice that goes along with the cell communication video. So this covers things like, GPCRs and, tyrosine kinase receptors, those kinds of things. So it's going to have both USABO and science bowl questions. So let's get started. So first we have our USABO questions, and we're starting with this one from the 2016 semifinals test, and this is from those that first section of questions, so you want to be going a little bit fast with these ones, and they're worth 1 point each. All right, so hopefully you had a little bit of time to do this one, so let's look at it. So essentially what we have here is just a normal GPCR system, except our GTP, we cannot turn it back into GDP in order to deactivate the G protein and thus deactivate, the enzyme that the G protein is acting on. So if you look at this diagram that shows how GPCRs work, essentially what we can't do is go to this last step where we had our GTP and we hydrolyze it to a GDP. So we're stuck perpetually in this step, and you'll see that means that the G protein's always active and the enzyme is always active. So if we look at all of these answer choices, the choice that matches this is answer choice D. The enzyme normally activated by the G protein will never inactivate. So if we look at all of the other answer choices, they're incorrect. So A says the GPCR's respective ligand will not bind it. That's not true because that's happening, outside of the cell. It's something completely removed from what's happening here. And then C says the inactive G protein will never be reactivated. That's not true. In fact, it's the opposite. The active G protein will never be inactivated. So yeah. And then E says the cycle of the GPCR will not be affected. We just established that it will. So, yeah. Now look at this one. So just pause the video, give it a try. And this one is from the second section of the semifinal exam from 2014. And this one's going to be multiple select. So select as many answers. All right, so let's look at this. So this one's going to take a little bit longer. Just because it comes from the second section of the semifinals test, it's worth twice as many points. So let's see. It asks where there's going to be amplification in this process. So you can kind of logic these out, and you can also just memorize kind of the general places where you get amplification. So I've got a diagram right here that shows that. But let's just see if we can do it without the diagram. So A says from GPCR to G protein, and yeah, there is going to be amplification there just because one GPCR can correspond to, multiple G proteins. And then B says adenylyl cyclase, this cyclic AMP, that's true. One adenylyl cyclase is able to convert multiple, ATPs into cyclic AMPs, before it's deactivated. And then C says cyclic AMP to protein kinase cascade. That's not true because one cyclic AMP causes one cascade. And finally, and D says protein kinase cascade. Yeah, so there's going to be a lot of amplification there. That's where the bulk of it happens. And then E says G protein to adenylyl cyclase. That's not true. There is no amplification happening there. So the answer is A, B, and D. So yeah, I feel like for most of those, it's somewhat intuitive. You can kind of just think, okay, is there not? But it could be nice to just know this chart a little, So yeah. Now we're moving on to science bowl questions. So, take a quick look at this one. Since these are science bowl questions, you should get them pretty much as soon as, you're reading the correct answer choice. Alright, so hopefully you took a look at this one. The correct answer is W. The G protein binds the ligand-bound receptor. So for all of these answer choices, they're all correct statements as to what happens in like a GPCR process, but W happens right after the receptor binds to the ligand. Because if you remember, that G protein is just kind of hanging on the membrane right there. It takes a ligand binding to the receptor for that G protein to go and associate with the receptor as well. All right, take a look at this one. Yeah, so for this one, it's a very easy like mem. So you just kind of had to know this one. It's Y7. You've got those seven transmembrane helices, and that's kind of one of the distinctive features of a GPCR. And then this one. Okay, so the answer for this one is W, and you can kind of go process of elimination. You know, the ion channel coupled receptor, that is a surface cell receptor, or a cell surface receptor. Same with tyrosine kinase receptors and GPCRs. So those three make up like the generally accepted cell surface receptors. So the answer is W, laminar receptor. And yep, that is it. I hope this was helpful for y'all guys. This is a pretty important concept to know well, whether or not, there's a lot of questions on it, because there are a lot of other biological concepts based on this one. So I hope this was informative, and stay tuned for the next video. Thank you.